The immune system's most impending function is to clearly identify self. That means every single cell and multiple biochemicals, mainly proteins, that will make you, you. You clearly understand that the interface between the cell and its external environment, that includes the interstitial fluids and any biochemicals that surround the cell, is the plasma membrane. If you were to place a self marker to label the specific cells within your body, where would you locate it? It has to be at the cell membrane. That is the plasma membrane. You have hundreds of thousands of molecules of phospholipids forming the membrane. That would not be a good option. The best option would be a protein that will be sticking out of the cell membrane and can easily be identified. This is mainly because it is three-dimensional. You will need to remember with clarity what the letters MHC represent. Major histocompatibility antigen. These antennas coming out of the cell plasma membrane are fabricated from your specific DNA and will belong only to you. Notice the word histocompatibility. Histo means histology, or the composition of the tissue, and compatibility to mean their similarity. These are the proteins that are cross-matched when an organ transplant is going to be executed. The whole idea is to prevent a massive rejection from a tissue that does not belong to you. When there is a particle, mainly molecules, that your immune system can recognize as foreign or non-self, it is designed to be treated as an adversary. Every time your adaptive immune system generates a response to this molecule, it will be known as an antigen. An immune response from the adaptive immune system can only be elicited by an antigen. So how do we describe an antigen? This beautiful diagram represents a scale model of the protein cover of a virus. This cover is named a capsid. The capsid, as you can see, is made up of multiple polypeptides. That is why each one is colored differently. Let's focus for a moment on the blue areas in this capsid. You can see that our diagram shows one right at the center. Peptides make excellent antigens because of their three-dimensional surfaces and the ability that our adaptive immune system will have to recognize them. The blue protein that I highlighted with the black line in the previous slide is represented diagrammatically in this one. I want to outline the blue region that represents the coiled protein. In this diagram, the whole viral capsid is represented as a diagram. It appears as a sphere on the right side of the slide. This means that if this capsid enters the fluids of our body and is part of our external environment, it will elicit an immune response. There is just one more concept that needs to be added. Notice the section that says times 12 next to the sphere. Notice the even smaller section that says times 60 on the far left to the sphere. Look back at the sphere and realize that if you go around the area designated by bold black lines, you will see the outline of the section that says times 12. This section alone, taken off the capsid of this virus, will be sufficient to elicit the immune response. We will call this section of an antigen an antigenic determinant or an epitope. If we go to the smaller section, now removed from the capsid, look at the area that looks like a triangle. Focus now on the section that says times 60, and what you have is a brand new antigenic determinant or epitope. This means that if a viral capsid enters a macrophage through phagocytosis and the lysosomes tear it apart, it can be broken into hundreds, if not thousands, of different antigenic determinants. Each one of them, as a separate fragment from the main antigen, will be able to elicit an immune response from our adaptive system. This is essential to be able to respond to a single antigen by accessing multiple antigenic determinants. The immune response, because of this, is significantly enhanced. To end, the blue coiled protein presented in the previous slides is the one I am highlighting at the center of the epitope, or the first antigenic determinant that we discussed. Antigens deriving from your own DNA and presented on your own cells by MHC1 molecules are called self-antigens. They are not immunogens or epitopes. An antigen and its epitopes will be recognized as non-self. One further category to present are very small antigens known as haptens. These molecules are not immunogens on their own. They will not elicit an immune response directly. Nevertheless, they can engage a carrier molecule within our interstitial fluids or the plasma of blood. A carrier molecule will usually be a protein. This will then produce a complete antigen. This new particle will be immunogenic. That is, it can initiate an immune response